All right, ladies and gentlemen, welcome back. We're going to be looking at the cell again, and specifically, we're looking at the plasma membrane, its structure, and how it will reflect its function. So, first of all, we name this the fluid mosaic model, and it's because, in terms of mosaic, that means that there's multiple components that are going to be constructing this membrane: phospholipids, proteins, and carbohydrates, as well as the fluid part just means that it's not static. It's able to move back and forth. There's some give in it. Okay, so let's get right into the phospholipids. The phospholipids, as you can see here, there was two layers. And I'm just going to flash back again to that last picture. As you can see, you have this little circular head and these two little tails coming off. That makes up one phospholipid, but there's a whole row of them here. And then right underneath it, you have this inverse picture right below it. So what each of those phospholipids are made up of is what we'd call a phosphate head and a lipid tail. If you remember your chemistry, phosphate this phosphate head will be polar, which we say is hydrophilic. It likes water. And these lipid tails are considered nonpolar or hydrophobic, and they do not like water. And so what this does is, because of this structure, it creates the necessity of these nonpolar tails be faced in together. What it does is it squeezes out all the water, and it creates a barrier between the aqueous or watery outside of the cell and the aqueous watery inside of the cell. Okay. If you look at it in terms of a little bit more specifics, you can actually see the carbon hydrogen arrangement. We have our lipid tails, are essentially variation of a glycerol and phosphate head, and then a polar substance or molecule that's going to be on top of it. Okay, so what this does for us, because of this barrier, there's only certain molecules that can actually get in without assistance. Since there's not a lot of room between, only really small and uncharged molecules or lipids can actually squeeze through on its own. But even then, it's only going to be with its own concentration gradient, which means it has to go from high amounts of substance to low, which is something we'll see in our next video. Okay. Now, the other component here is going to be the proteins. So within, and I'm just going to go back again just for a second to bring up this one. As you can see, there's multiple other blue components here. These are all going to be proteins. Okay, some of them completely bridge the entire membrane, and some of them are only located on the inside, which we would call these peripheral membranes. So the first protein that we're going to see here is called a channel protein. And as you can see from the picture, it allows substances to travel through that weren't going to be able to get through on their own because either they're too big or they're charged. So they're still going to be going with its own concentration gradient, or from high concentration to low, but it just needs these proteins in order for them to pass through. So these are called channel proteins. The next one are called carrier proteins. And these carrier proteins are going to physically grab on and hold and bring through a molecule. Once again, it can be because they were too big or charged, or a lot of the times it's because it actually has to go against its concentration gradient. They can't just open up a door and allow something to pass through, because it always goes from high to low. So instead, what they want to do is, if you want to be able to force something against its concentration gradient, you need a carrier protein. Okay, an example of this would be glucose in your kidney. We want to make sure all of the glucose that we've filtered out go back into our blood and none leaves in our urine, because it's so important as an energy molecule. And here's a little clip of a carrier protein. Specifically, this one's a sodium potassium pump. So as you can see, we have our sodium Na plus molecules that are moving into our purple protein. ATP is going to allow it to change its shape, which then allows the sodium to pass through to the other side. So it goes from the inside to outside. It needs this carrier protein because it is going against its concentration gradient. Now that the protein is opened up to the other side, it also allows for the next molecule or ion, potassium, to come and attach. This attachment causes the phosphate to get kicked off, which is then going to change the shape of the protein back to the original shape. Potassium could enter the cell, and once again, sodium is going to be able to enter and start this process again. The main takeaway here is that in both of these scenarios, for sodium and potassium, they're going against their concentration gradient. So they're going from low to high concentration, and that's why ATP, or the energy molecule, is going to be needed. The next one is a receptor protein. We're not going to do a ton with this until we get into the digestive system. We talk about insulin and blood sugar regulation. But the example here is that this is a receptor protein that is on the surface of the cell membrane, and it allows for a molecule to join to it and that has a signal. And what that signal is going to do is it causes a bunch of things to happen in your cell and ultimately lead to some cellular response. In this example, the cellular response is going to be creating a transcription factor to allow protein synthesis to occur. The next one, once again, we won't do a lot with this in this course, but you will see it if you continue on in biology, would be using an enzymatic protein. This is on the inside of the cell, and it just allows it to change one molecule into the next. So it catalyzes it or speeds up a chemical reaction. In this case, it catalyzes the reaction of ATP, which are energy molecule, into cyclic AMP, which is also known as CAMP, and it's a signaling molecule in your body. And then the last one is cell recognition proteins. This is actually a protein, but it also has a carbohydrate group. And depending what that carbohydrate group is, will give your cell a specific identity. So this is how your blood type differs from somebody else. A type blood will have a different signaling carbohydrate on the protein than B type blood. And then AB will have both the A signaling carbohydrate and the B, whereas O is something we'll see in circulation. The circulation unit doesn't have the signaling carbohydrate on the proteins. Alrighty, so that's it in terms of our structure. We have to really closely look at what is the impact of the structure, specifically the phospholipids and the proteins, and how does that allow the cell membrane to have a specific function. More importantly, what is able to pass through on its own and what is going to need some assistance.